Today we'll be discussing the genomics of bacterial host adaptation and how it works, but first we need to have an understanding of what it entails. Bacterial host adaptation is defined as the ability of bacteria to be transferred from one host to another. An example of this can be seen from the transfer of the well-known human pathogen S. aureus from humans to chickens. The DNA within the bacteria undergoes certain changes through mutations and other processes that allow it to adapt to the environment of the chicken. However, this transfer can be fatal. It is important to understand how this process works, as it is estimated that 60% of all known human pathogens originated from other animal species, and having an understanding of the process could help to develop a prevention. Further, through globalization, it is now more important than ever to be able to control the transfer of pathogens between hosts, as new species are being exposed to many new bacteria from all around the world that could carry with them deadly diseases. Gemma Langridge's 2015 study compares the genomes of the Salmonella enterica lineage in order to identify the differences between the genomes that are responsible for specialized host adaptation. Within this report, different pathogens that were closely related were studied, one found in multiple hosts including humans, one found in bird species, and one found in cattle species. It was largely found that host adaptation occurred as a result of multiple events including pseudogene formation, gene acquisition, as well as gene decay. Pseudogenes are defined as a gene harboring a mutation relative to an intact version of that gene, or more simply, genes that have accumulated enough mutations to render them non-functional. Gene decay has been found to be a common feature of bacteria undergoing adaptation to new environments, and it describes the genome that has undergone extensive changes and loss of gene functions, often in the form of pseudogenes. Langridge's study found that the pseudogenes towards the tip of the chromosomal phylogenetic tree were found to be related to the membranal surface structures, which is strongly related to the survival of a cell within a new host. This suggests that over time the bacteria have lost key functional genes that account for their membrane, making them reliant on life within a host, and that different pseudogenes can be connected to different hosts. This leads to the well-specialized bacteria evolved from one common ancestor that are perfectly adapted to their new respective environments. This is further supported through Guinain's study of the Staphylococcus aureus strain that found changes in cell wall-associated proteins in bacteria of different hosts, which exaggerates the importance of these genes in pathogen-to-host interactions. Gene acquisition or horizontal gene transfer describes the transfer of genes within a generation rather than through generations. A study conducted by Catriona Guinain explored the evolution of host adaptation of Staphylococcus aureus, a notorious human pathogen, and it was found that horizontal gene transfer was a leading factor in the process of host adaptation that allowed the bacteria to spread to many animal species, including cattle species. As a result of horizontal gene transfer, bacteria containing beneficial mutations can spread, creating a population that is well adapted to a new host. A whole genome sequence was performed for both human and bovine or cattle strains, which resulted in the discovery of considerable variation among genes encoding for proteins that account to adherence, toxin productions, metabolism, replication and repair, as well as gene regulation. This suggests that these functions play a role in the process of host adaptation, and that a change in one of these genes can specialize the cell to a new host. This horizontal transfer has been proven to have significant relation to greater interaction between new hosts and the bacteria. The ability of bacteria to adapt to new host environments poses a great threat to the safety of all organisms, as it permits the large-scale spread of pathogens around the world, largely as a result of globalization. By understanding that this ability is largely as a result of the formation of pseudogenes, often related to the cell membrane or cell wall, as well as the process of horizontal gene transfer that aids the spread of beneficial mutations, we have gained an insight into the process of host adaptation, and it is now possible to manufacture a prevention or to utilize the process for research purposes.